Okay, hello everyone. I am Dr. Kushal Salunke. Now, in this practical demonstration, we will be covering a color reaction of proteins. Okay, so proteins are made up of larger number of different alpha amino acids, which will be joined together by a peptide linkages. And all the protein on the hydrolysis with strong acids or enzymes, they will yield amino acids. And presence of different functional groups in various amino acids gives a different color reactions of protein. Okay, so why proteins are yielding the color reaction, or why the proteins are giving the color reaction? Because proteins has different amino acids, and these different amino acids have a different functional group. That's why this will give a different color reactions. Okay, so first test, you can see this is a biuret test. Okay, so what is the method of biuret test? So you have to take 4 ml of 5% of NaOH. On it you will be adding 3 drops of 1% of copper sulfate. You have to mix it and you have to divide in 2 parts. To one part you are adding a water and you have to mix it well, and to another part you have to add a given test solution. Okay, on my left hand side you can see the test is positive, it will yield violet color, and on the right hand side you can see the test is negative. Okay, because I have added the water, and water is a protein free. Okay, because so left hand side is test and right hand side is control. Okay, so there is no change in the color on a control side. What is the inference? Inference of biuret test is 2 or more peptide linkages are present. Okay, so either 2 or more peptide linkages are present, that's why the protein is present. And what is the principle of biuret test? Why the purple color is developed? This is going to be developing because the formation of copper coordination complex between cupric hydroxide and peptide linkages of protein. That's why it will yield a purple color. So this reaction is due to presence of peptide linkages. Hence all the proteins will show this reaction is positive. Okay. Color intensity will depending upon the presence of number of peptide linkages. More and more dark color will be proportionate to more and more number of peptide linkages. So insoluble proteins like keratin and collagen does not give this test. Important point to note, collagen and keratin, they are insoluble proteins. It will not going to yield this particular test. Okay, I hope you understand the biuret test clearly. On second hand, you can see this test. This is a ninhydrin test. How will you perform this test? You have to take 2 ml of protein solution given to you, plus you have to add 4 drops of 1% of ninhydrin solution, and you have to mix it and you have to boil it for 1 minute, and it will result in a dark lavender color, which indicates free alpha amino acids are present. Free alpha amino acids present. This test indicates free alpha amino acid is present. Okay, so this is ninhydrin test. What is the principle? So you know ninhydrin is a powerful oxidizing agent. You have to answer this in a viva. When the examiner asks you, tell me the principle of ninhydrin test, you have to add this word. Ninhydrin is a powerful oxidizing agent and causing oxidative decarboxylation of free alpha amino acid, producing aldehyde and ammonia. So the reduced ninhydrin called hydradentine then reacts with this ammonia, and another molecule of ninhydrin will produce a bluish color coordination complex. This is the result of which they will develop a lavender color. Okay, you will be asked this question in your viva. Why ninhydrin will show this purplish blue color or bluish purple color? Okay, I hope this thing is clear. Inference will be free alpha amino acid is present. So next in a line you will be seeing this xanthoprotic reaction. Okay, xanthoprotic reaction will be specifically for amino acid which contains the aromatic compound, aromatic amino acids. Okay, so you have to take 3 ml of solution, plus you have to add carefully 1 ml of concentrated HNO3 nitric acid. Okay, and you have to boil it for 1 minute. So after boiling you have to settle it down and let it be cooled for another 1 minute, and then you have to add 2% of 2 ml of 40% of NaOH, and you have to mix it well. So observation, you can see this test will develop a yellow color. Okay, and this yellow color is deepens into the orange color, I'll show you the first part of this test. Okay, see this is the first part of the test on my right hand side. After boiling and before adding 40% of NaOH, you will be seeing this kind of reaction on my right hand side. And once I add 2 ml of 40% of NaOH solution, this will deepens into this kind of yellow color. So this test is positive in my left hand side. Okay, so inference will be aromatic amino acid is present like tyrosine and tryptophan, and phenylalanine is present. Okay, so principle of xanthoprotic test, many a times they will ask in a viva. So aromatic amino acid, tyrosine, tryptophan and phenylalanine respond to this test due to presence of benzene ring. There is a nitration of phenyl group of aromatic acids on heating with concentrated HNO3. So benzene or phenyl group will be going to be a nitration process by we will be heating the HNO3. And the nitrophenyl group gives a yellow color due to the solution, which turns into orange due to ionization in alkaline medium. So why this? So this color is due to, this yellow color is due to nitrophenyl group, okay, which is yielding a yellow color. You can see the yellow color precipitates. When I will be adding the alkaline solution, this will going to deepen into yellow color, more yellow, bright yellow color, dark yellow color, because of ionization. This test is positive. I hope this thing is very clear to you. So now next in a line, you can see this is a Millen's test. What you have to do for this test, you have to take 2 ml of protein solution given to you, and then you have to add 1 ml of 10% mercuric sulfate. You have to mix it well, and then you have to boil it for 1 minute. And after boiling, you have to be let it cool down. And then after cooling down, you have to add 2 drops of 1% of sodium nitrate solution, and again you have to warm it gently. And this test, on the observation, you can see the red color precipitate. You can see this particular precipitates on the top, and few precipitates are on the bottom also, which is floating. You might not see it. Okay, and the inference of this particular test is hydroxyphenyl group is positive. So what amino acid will contain hydroxyphenyl group? That is tyrosine. Tyrosine amino acid is positive in this particular case. So what is the principle of Millen's test? Okay, so this test is answered by protein containing hydroxyphenyl group, that is a tyrosine. So the phenol group of tyrosine which gets exposed on boiling with nitrous acid, okay, to form a mercuric phenolate compound, which will give a red color on precipitation reaction. So mercuric phenolate is a reason for this red color development. So next in a continuation, we will be having this test which is called as aldehyde test. Hope you can see the aldehyde test. Okay, so aldehyde test, for this test you have to take 2 ml of protein solution. On it you have to add 2 to 3 drops of formalin, which is diluted formalin, and on the top of it you have to add 1 ml of mercuric sulfate, and then you have to mix it and mix it well. Then slowly you have to add 2 to 3 ml of concentrated H2SO4 carefully by the side of test tube, and you can see this test. The violet colored ring is formed at the junction of 2 layers, very nicely formed violet colored ring at the junction of 2 layers. So this test is positive, and this will indicate the indole ring. So the tryptophan is positive in this particular test solution. So principle is indole ring of tryptophan undergoes oxidation by mercuric sulfate. So oxidizing agent here is a mercuric sulfate with sulfuric acid, and the formaldehyde will form, and this will react with the oxidized product of indole molecules to give violet color complex. So why violet color complex is formed? The oxidized product of the oxidation with this particular indole ring and will give a violet complex color. So also known as Hopkins test, Hopkins-Coles test. Okay, so tryptophan is positive in this particular case. So now on the next, we are observing this test. You can see the sulfur test. Okay, you can see in this test there is a brownish color tinge. You can see the slightly brownish color tinge, and sometimes the blackish color precipitate also seen. And this sulfur test is mainly, as the name suggests, it is for sulfur containing amino acid, that is cysteine and cysteine. Okay, methionine does not give this test Okay, I know all of you having the question. Sulfur containing amino acid is methionine and cysteine, but methionine won't give this test, and why we will be seeing in the subsequent part. So you have to take 2 ml protein solution plus 2 ml of 40% NaOH. You have to mix the content well and you have to boil it for 2 to 3 minutes. Let it cool down, and then you have to add 2 to 3 drops of lead acetate solution, and again you have to mix it. And you can see this inference: black or brown color precipitate. In this test, brownish color precipitate is seen, but that is due to sulfur containing amino acid. So sulfur containing amino acid cysteine or cysteine is positive. Okay, closely you can see the brownish or blackish color precipitate is present. Okay, so when a protein containing cysteine and cysteine is boiled with strong alkali, here we are adding 40% NaOH. So 40% NaOH is strong alkali. The organic sulfur is converted into inorganic sulfide, and addition of lead acetate to this solution forms a black color precipitate of insoluble lead sulfide. So black color precipitate is due to lead sulfide. Methionine does not answer this test due to presence of thioester linkage. So methionine mein there is a thioester linkage which does not allow the release of sulfur, because thioester linkage, sulfur is in the middle surrounded by 2 methyl groups. So methyl groups does not release this particular sulfur, and the methionine will not show a reaction positive. So on subsequently we are seeing the Sakaguchi's test. Okay, so you have to take 2 ml of protein solution plus 2 drops of alpha naphthol, you have to add on it, plus 1 ml of 5% of NaOH. Okay, then you have to mix it. Now on the top of it you have to add 3 drops of sodium hypochlorite solution and mix it well. You can see very nicely formed carmine red color is formed. Okay, so this test is positive for guanidine group, and guanidine group containing amino acid is arginine. So principle of this test will be: in alkaline medium, alpha naphthol combined with guanidine group of arginine to form the complex, and this complex is going to be oxidized by our oxidizing agent sodium hypochlorite, and this sodium hypochlorite oxidation to form bright red color complex. You can see very bright red color complex. Okay, in the background, white background, you can see this very bright color complex is formed, and this test is Sakaguchi's test. Okay, this is Sakaguchi's test. This test is positive. Remember, we can keep this test in your spotting examination, so all of you must understand this test. Okay, so now we are observing the Mollis test. Okay, you have to ignore this green color. Okay, this is due to contamination of sulfuric acid. You have to ignore this green color, but you can see very nicely formed purple violet color ring at the junction of acid and the solution. Okay, above one is solution, and you can see bottom part is acid. So this test is nothing newer to you. You have performed this test in your carbohydrate practical also. So all the carbohydrates, when treated with concentrated sulfuric acid, undergoes dehydration to form perfural derivatives, which is on condensation reaction with alpha naphthol will be forming a violet colored complex. Now how will you perform this test? You have to take 2 ml of protein solution plus 2 drops of alpha naphthol in it, and you have to mix and add 2 ml of concentrated sulfuric acid carefully through the side of test tube without shaking, and you can see the purple violet color ring is formed between the acid and solution. Glycoprotein shows this test positive. Okay, some of the protein, example albumin, contains the carbohydrates like glucose, galactose as a prosthetic group, so this test is also positive for that particular protein. Okay, thank you. So I hope this practically is very clear to you. We keep this test in the spotting examination. Okay, I hope everything is very clear. If you have any doubt, you can ask me. Okay, best of luck for the examination and happy reading. Thank you.